bioinformatics, some of the computer applications that we can use to study genes and gene products. So bioinformatics is typically used to analyze genome data using computers. It generates lots of information on genome content and structure and protein function and structure as well. We can also annotate genes and learn about their position in the genome or what they even do without even doing any wet lab work at all. As far as genome annotation goes, one of the things we can do is locate genes in a particular genome map, and I'll show you that an example of that. We can identify open reading frames, which again is just another fancy way of saying genes in a genome, by looking at reading frames of greater than 100 codons that are not interrupted by a stop codon, for example. So computers are, quote unquote, computer software is, quote unquote, trained to look for these open reading frames that might be possible genes. And so that gives us the ability to hone in on hunks of DNA that might be interesting. Then we can grab those little pieces of DNA that are not interrupted by stop codons and use other applications to identify the possible gene. We can also look for ribosome binding sites and terminator sequences that flank long stretches or open reading frames as a way to determine whether or not they're potential genes. So one of the things that we use, one of the most basic, and I'm sure most people in this room have used, is DNA sequence analysis using BLAST. And so BLAST is great for if you have a hunk of DNA of unknown annotation to potentially identify that. And so again, I happen to have a DNA sequence handy here, and so again, I've annotated some features of this particular gene, so of this particular hunk of DNA. And so, for example, here's a putative start codon ATG. There's another one just upstream of it as well. But here's but odds are and we can figure out which one is the actual start. But anyway, so here's a putative start codon. Here's a putative stop codon, and so and these other highlighted regions are just PCR primer sites. But anyway, we can I'll just take a bit of that. And then go to the BLAST, and so BLAST will allow us to do a lot of things. Like we can input amino acid sequences. We can do a translated nucleotide to protein. We can do protein translated to nucleotide. But this is just a straightforward nucleotide BLAST because we have our data is DNA, and so real easily we just pop that right into the box. Usually you don't have to mess around with these parameters here, but if you feel like the DNA sequence is not very good, high quality, you can change some of these parameters, and you could also vary the database that you're using. But I usually just use nucleotide collection. Or you could even enter the specific organism, because I know that this hunk of DNA came from Vibrio fischeri, so I can narrow my search. But the computational power is so great, it's pretty darn quick regardless. You could probably slow it down by a few seconds. I'm sorry, speed it up by a few seconds by putting a specific organism in there. But anyway, long story short, we can just go through and BLAST that. And the output is what we would expect, Vibrio fischeri gene. And so again, and this comes to us with a score. These are all statistical values. You don't need to know these, but how the scores are computed. Query coverage just means that your sequence aligned up 100%. So that's percent identity here. There was 100% identical match across the entire 100% sequence that I input. And so this E value here is basically just a measure of how likely this is to be a random match. And so again, the closer you are to 0, the stronger your DNA sequence is. So here I'm 0. So we know that this thing is a 100% match. And you can look specifically at the alignment to show that. So here's my query sequence is on top. The subject sequence from the database is on the bottom. There's it was 1,080 bases long, 100% match. No gaps, and it's a plus plus, so that it's on the same strand. And so again, this is a a great way to get feedback on the genome sequence. Now, more specifically, if I want to, in the case of, there are different databases that you can use. And so I use KEGG. And so KEGG is one that gives you a ton of information. And so I can do, for example, a KEGG BLAST, and I can put my sequence in there. And then what I can do is, like I was telling you before, I'll say BLAST N, and I'll put my favorite organism in, because I know it's from Vibrio fischeri, so I'll put the abbreviation for Vibrio fischeri, which is VFI, and that tells me what the gene specifically is. Now, instead of just saying, okay, this thing's in Vibrio fischeri, and here's the genomic region, KEGG will tell me specifically what it is, and you can see that, and then I can click on that and go right to this information page about the particular gene. So here's the annotated entry. Here's the definition, or the annotation definition of the organism, of the gene, the organism it comes from, and cool things like motifs, for example. The software will give you a sense for what motifs might be in the hypothetical protein that this gene encodes. So again, you see this really strong endonuclease phosphatase family domain right here in the protein. This bar represents the amino acid sequence translated from the DNA that I inserted, and so it gives you these really precise estimates of different domains. Here's the amino acid sequence. Another cool feature is the genome map. And so here's the gene, and here's other genes that it sits next to. This is just a fragment of the genome. Obviously, I can go in different directions and see different regions, but you can see potential operons that the gene sits next to. You could even see if this gene was in an operon. It doesn't look like it's in an operon, right? Because there's no other genes going in the same direction. But anyway, the point is that you can get just a huge amount of information. And here's the coding sequence of the gene, starting with ATG and ending with the stop code on TAA. And then if I ask for additional bases upstream, 200, 100, whatever, or downstream, I can look for promoter elements as well. So if I ask this thing to say, oh, give me 150 base pairs upstream. So here, I could dig around and look for potential TATA boxes. I could look for potential ribosome binding sites, -35 sequences, and so forth in the promoter. There are open reading frame finders that you can use, for example. So if a giant hunk of DNA, I could load that in here, and then the output would tell me, okay, you have a possible open reading frame in this location. So there's really some really cool tools for that. As far as functional genomics goes, it's a method for determining how the genome works, and it uses physical maps of the locations of genes, but it potentially provides information on metabolic pathways and transport mechanisms, regulatory and signal transduction mechanisms. And so you can, without even doing any wet work, you can get a full genome map of an organism based on all the DNA sequencing, and then without any additional wet work, you can construct functional maps to understand the physiology of particular organisms. Now, I'm not going to ask you to memorize anything here, but you can see how sophisticated these things can be just off of the DNA sequence. We know, for example, that this organism has ribose-galactose transporters, and so obviously it seems like it makes sense that the organism can use ribose and galactose. It has specific galactose transporters. Over here, there's cation transporters. And so again, you see all kinds of different aspects of the physiology of an organism without lifting a single pipette. So again, as I've mentioned, some of the information that you can get from KEGG, genome location and motifs, I've shown you all that. You can get information about the hierarchy and so what other organisms have something similar to this. You can also, if you look at the if you look at the go back to here, this is what I wanted to see, the pathway. If you click on pathway in KEGG, the KEGG will give you different pathways that this thing might participate in. Again, super important information that you can generate from a particular gene without even lifting a pipette. Another method that we use in molecular biology is qRT-PCR, or different versions of PCR that are more sophisticated than just good old-fashioned endpoint PCR. In this particular case, it's a gene expression tool called qRT-PCR, quantitative RT-PCR. In this particular chemistry that's illustrated, it's TACMan. And so the basic gist of it is that you have a forward primer and a probe with some kind of fluorescent marker and a quencher, a DNA template to analyze, and a reverse primer. And so in this particular case, you denature your templates. And so again, what you're trying to do here, I should back up and say, what you're trying to do here is quantify gene expression. So in this case, the DNA. So you start with a RNA sample, for example, and asking the question, how much of a particular species of messenger RNA is in a complex sample after an organism has been, for example, grown under a different circumstance, like low iron or high iron. And so you can take the low iron environment, for example, and extract messenger RNA from that, and take the high iron example or sample and extract messenger RNA from that, and say, okay, what's the influence of iron on expression of a specific gene? And so your specific gene primers and your specific gene probe can be inserted along with the DNA template, and then you would denature and you would anneal the forward primer, you would anneal the probe, and basically as the forward primer moves through and knocks the fluorescent dye off of the probe, a fluorescent reader in the thermocycler would detect this. And so every time this happens, the fluorescent reader records it and plots it as a function of cycle number, PCR cycle number. And so this animation is really useful to learn about real-time PCR. You even get to see an instrument. We used to have one of these, but I think we've got something a little bit more sophisticated now. But anyway, if you watch this particular video on this particular YouTube video, you can get a good sense of how the real-time PCR quantitative approach works. I should also mention that Fisher has a really good, and so again, if you take this URL here, this is Thermo Fisher, and they talk about how the TACMan assay works to take you right through the whole procedure. So again, I do want you to be able to understand that and be conversational about that and answer some exam questions about it. But the output looks a little bit like this, where you have an amplification curve with fluorescence emission on the y-axis and cycle number on the x-axis. And so during the initiation phase of the reaction, there's PCR happening, and so basically this is happening where the polymerase and the primer, the primer anneals and the polymerase goes through, and it's knocking fluorescent tags off of the probe. But it's not enough to move above this sort of background threshold. But eventually, after a certain number of cycles, and depending on how many copies of the particular messenger RNA which was converted to DNA are present, the CT or critical threshold will be reached, And so again, this is the point at which the values start becoming significant and rise above background. And so eventually you get a nice rise in fluorescence. And again, you can use the CT value as a way to figure out which gene is present in higher concentration. It shouldn't be surprising that if there's more copies of a particular gene in a QRT-PCR reaction, then your CT value will shift to the left, meaning it'll take fewer cycles to rise above the threshold because there's more copies of that particular um, piece of DNA. The slide here is just showing you another version of gene expression tool called the microarray. So in this particular example, microarray is used to analyze total RNA expression in a cell under different circumstances. Or in this particular case, it's two different cells, and you're just comparing messenger RNA production. And so in this hypothetical example, you have a normal cell versus a cancer cell. And then so somebody will then extract total messenger RNA from this cell type and total messenger RNA from this other cell type. And then the messenger RNA can be converted to cDNA. And so this workflow right here, the messenger RNA extraction and cDNA extraction, is something that we would use here too. We would start with messenger RNA extracted, and then we would do a QRT-PCR using the TACMan on cDNA or complementary DNA that was made from the RNA, just as you see here. The cDNA can be labeled, and so you can use a green fluorescent probe for the normal cell and a red fluorescent probe for the cancer cell. So in terms of bacterial genetics, you could do, instead of normal cell, it could be bacteria, in an infection versus bacteria growing on a TSA plate. So you could do any number of different kinds of experiments that you can think of. This all begins with, you can't do this without a gene chip. And so in this particular case, you have a gene chip that represents, and these things are tiny. These, it's like the size of a postage stamp on a regular glass slide. And the postage stamp that I alluded to was basically just spots containing each single-stranded DNA in the entire genome of the organism. So a single-stranded copy of each gene in the organism's genome. So here they only show you a 4x3 matrix, but again, imagine it's 3,000 gene genome, or whatever the organism is. Some are bigger, some are smaller. But the point is that just for clarity, they only show you 12 genes here. But then you would hybridize the probes, in the same way you would flood a slide with Gram stain reagents, crystal violet. You'd flood a slide with a pool of cDNAs from each condition, and let them hybridize to the wells, and then you can wash that off and then scan it on a microarray reader. And so so basically the legend here hopefully will help you walk through it. So in this particular panel, the gene that's here, nothing bound to it. And so that tells you that the messenger RNA wasn't present in the cells under the conditions of the lab. Here, because it's yellow, that's an intermediate between red and green, that tells you that equal numbers of red and green probes bound to the DNA sequences in that particular spot. And so it turned up yellow. So we would say that the RNA was present in both cells. However, what we're looking for is significant differences. And so anything that's green, for example, tells us that the messenger RNA was only found in that particular condition. And so here's an example. Whatever this gene is right here, we know that this gene is active in the normal cell and not active in the cancer cell. And that's significant. Whereas this red one is the opposite, where that would be like, for example, this, which is this here, which is represented, tells us that there's messenger RNA present in the cancer cell, but not in the normal cell. So the machine basically sees this, And what it will do is then interpret this information for you as, relative levels of expression. So rather than just saying green, red, yellow, it would say specific numbers, like how much more green than red. And so, the output might be like, 200 units of green, zero point 0.1 units of red. And then you would know that this is a gene that's expressed in normal cells only. I'll post an animation for this microarray tool. The last thing I wanted to bring up was gene expression studies using proteomics. And so this is the iTrack workflow. And Dr. Tomanek has the environmental proteomics lab. And so, a lot of the proteomics that we do is through his lab. And so he has a mass spectrometer and different devices, HPLCs and MALDI-TOF, MS analysis, or MALDI-TOF, TOF, all these weird little names. But the bottom line is we typically use a mass spec to figure out protein identification. So it's the same concept as the microarray in that we can use different cell types, or even the same cell type growing under different conditions, or different cell types growing under the same conditions. You name it, sky's the limit. But the bottom line is you have your samples and you extract total proteins from those samples, and then they can be labeled differentially. And then that pool of proteins can then be subjected to HPLC and mass spectrometry to figure out what they are. And so you get a list of all the different proteins in the individual samples, but you would also get a relative expression level of each protein in the samples. And that will allow you then to figure out how much of a particular protein is being produced by a particular organism under a particular set of conditions.